In 1565, Bartholomew Dye was born in the town of Ware, Hertfordshire. Through burial registers and parish records, we can trace a modern lineage directly back to him, locating a specific point in 16th century England, where the family history begins. But the trail stops there. For most of our history, if an ancestor wasn't recorded by a church or a tax collector, they disappeared from the timeline. This is the paper wall, the point where traditional genealogy runs out of written evidence. Breaking this wall requires the Y700 Big Y test. Y700 is a high-resolution genetic test that sequences nearly the entire Y chromosome to identify ancestral markers passed from father to son over thousands of years. This methodology tracks the movement of entire populations across millennia, using shared mutations as geographical markers, rather than searching for individual names. These specific genetic markers persist long after the families who carried them were forgotten by history, providing a biological record of a lineage's true origin. If we move the timeline back from the 1500s to the Iron Age, we find that the genetic matches for this line appear in medieval Belgium and La Ten-era France. This creates a geographical puzzle. How did a lineage firmly rooted in continental Europe cross the English Channel to become a British family? One possibility is the Celtic expansion. This map shows the Hallstatt and La Ten cultures moving from their Central European core into Gaul and eventually the British Isles during the Iron Age. Another path is the Roman occupation. The Roman legions utilized auxiliaries from the Alps and Gaul, territories where this DNA is common, and stationed them across the dense road network of Roman Britain. A third route involves the later Germanic migrations, as Anglo-Saxon settlers moved into England from the Netherlands and Germany. While we cannot identify the specific vessel, the data shows this lineage arrived during the large-scale population shifts that established the genetic foundation of Roman and medieval Britain. Jumping back to 1300 BCE, we leave the British Isles entirely. The closest ancient matches appear in the Urnfield culture of Neckarsulm, Germany. The Urnfield culture, in purple, dominated the Upper Danube and Bavaria. This region contains the highest concentration of modern genetic matches for this line. This suggests the population wasn't merely passing through, but held territory in Central Europe for many centuries. Long before this family line became British, it was a permanent fixture of Bronze Age Germany. The origin point occurs around 2100 BCE with the formation of haplogroup RU152. A haplogroup is a major genetic branch sharing a single common ancestor. The birth of this genetic branch is linked to the Bell-Beaker culture, which was expanding through the Bohemia and Bavaria regions at that time. The Bell-Beaker people were pioneers of the early Bronze Age, identified by their distinct metalwork and intricate bell-shaped vessels found in their burial sites. Ancient skeletons recovered from Central European burial sites perfectly match the genetic code of this modern lineage, providing a direct link to the ancient data. The Dye line began as Bell-Beaker pioneers, standing in the center of Europe 4,500 years ago, long before the 16th century records of Hertfordshire. Tracing this lineage reveals an unbroken chain of survival, from the Bronze Age in Bavaria, through the Celtic and Roman eras, and finally into medieval England. This redefines the family tree. It is a biological map tracking how a population survived and moved through thousands of years of history. Ancient history didn't just happen around these ancestors. They were participants in those continental shifts. The same genetic markers found in Bronze Age warriors and Roman auxiliaries are carried in the cells of their descendants today. Every generation of survival, from the Bronze Age to the modern day, is encoded in the DNA of those living today.
We spend our lives looking forward, but our biology is an unbroken chain stretching backward into deep time. Today, commercial tests like the Big Y700, which sequences thousands of markers on the male-specific Y chromosome, to build a highly detailed family tree, allow modern individuals to link their DNA directly to ancient archaeological excavation sites. But when your results show an exact genetic match to a 4,000-year-old skeleton found in a Bronze Age grave, you haven't necessarily found your direct great-great-grandfather. What you have actually found is a collateral relative. You and that ancient skeleton sit at the ends of different branches that converge upward on a single unseen common ancestor who lived much earlier. To trace a deep historical lineage like the European RU152, we have to abandon the idea of tracking a single family's cross-country road trip. Instead, we have to map the shifting centers of gravity for entire populations. To avoid historical illusions, personal genealogy requires the same rigorous statistical modeling used by population geneticists. This is a macro view of the human Y-DNA phylogenetic tree. The mechanics are straightforward. Every time a genetic mutation occurs, it creates a new branch. Every man on that new branch shares a common paternal ancestor exactly at that specific splitting point. This chart tracks the specific descent of R1b, the massive Bronze Age lineage that acts as the primary genetic trunk for much of Western Europe. In genetic terms, a haplogroup is simply a large population group of people who share a common ancestor on the patrilineal or matrilineal line. If we follow the R1b trunk downward through the Bronze Age cascade, the population continuously splinters into more highly specific regional groups, eventually reaching a major node designated RU152. This detailed tree shows the U152 architecture. By continuing down through the Alpine Celtic and Italic branches, we can isolate the exact position of a specific modern testing microbranch, like FT383849. The genetic estimates place the formation of the parent U152 node around 2300 BCE. The smaller FT383849 subbranch splits off shortly after, around 2100 BCE. This simplified structure of the U152 tree illustrates a critical concept. A specific branch like FT383849 is not an independent origin point. It is a deeply nested horizontal subset of a broader European population. A personal haplogroup designation provides a highly specific coordinate within a massive, interconnected human population structure. To ground these genetic branches in physical geography, researchers rely on hard data from large-scale archaeological excavations. Scientists extract DNA from the bones and teeth found at these sites, sequence it, and map it directly onto the modern phylogenetic tree. When an ancient skeleton matches your branch, it acts as a proxy. Think of it as a V-shaped graph. One line ends with the modern tester, and the other line ends with the ancient skeleton, both converging upward at a shared ancestor node. In the reality of the Bronze Age, multiple men carrying this exact lineage existed simultaneously across different villages. Because populations expand horizontally, the individual found in a specific grave could easily belong to a neighboring tribe, rather than being your direct forefather. What an ancient DNA match provides is definitive physical evidence that a specific lineage was present in a specific location at a given time. These ancient samples serve as geographic anchor points indicating where a broader population lived. They are not waypoints mapping a single family's historical migration. Ancient DNA samples are sparse, unevenly excavated, and scattered widely across the European continent. Taking a simple, equal average of these physical locations is mathematically dangerous. One random outlier sample found far from the core population could pull the estimated origin point into entirely the wrong country. To solve this, researchers use a statistical modeling solution known as a weighted average. The algorithm evaluates 4 variables. First is genetic closeness. Exact subbranch matches exert a much stronger geographic pull on the calculation than distant parent branch batches. Second is temporal closeness. Samples carbon dated closer to the exact time the specific node formed, such as 2100 BCE for our Bronze Age branch, carry higher weight. Third is sample density. Clusters of multiple noching samples excavated in one concentrated region naturally compound their gravitational pull. Finally, data confidence. High-coverage genomes with confirmed archaeological dating easily outweigh fragmented reads with uncertain timelines. When we run this formula for the early U152 lineage, it generates a secure statistical probability zone, strongly centered in the rolling hills and plains of Bohemia, Bavaria, and the Upper Danube.
This center of gravity calculation replaces the illusion of a pinpointed GPS coordinate with a rigorous, mathematically defensible regional homeland. We have to contrast that ancient calculated center of gravity with the geographic distribution of modern individuals taking big Y700 tests today. A naive look at a modern DNA database might make a specific branch look entirely British, simply because all the present-day matches happen to live there. The root cause of this illusion is market demographics. Commercial DNA testing is highly accessible and popular in the United States and the United Kingdom, but it remains much rarer across Central and Eastern Europe. An absence of modern DNA matches in Eastern or Central Europe does not mean the lineage was historically absent from those regions. That absence merely means the men currently living in those areas haven't swabbed their cheeks and uploaded their raw data to a commercial database yet. Modern testing clusters only show where a lineage's descendants survived and had the means to participate in genetic genealogy, not where the lineage originated. This map illustrates the Urnfield culture covering Central Europe. By synthesizing ancient proxy data with the weighted average model, we map the probable migration of this population. Over hundreds of years, that calculated center of gravity slowly moves westward. For lineages like RU152, the broad trajectory starts in Central Europe, pushes steadily toward the Rhine and Gaul, and eventually crosses the Channel into the United Kingdom. This represents the gradual, generational spread of a massive population group. It is a demographic shift, not the specific travel itinerary of a single family line. As archaeologists excavate new ancient skeletons and modern testers add new data to the pool, this statistical center of gravity will inevitably shift and refine itself again. Genetic genealogy is not the discovery of absolute historical certainty. It is the continuous, rigorous refinement of the deep probabilities of our human past.